Okay. Salam sejahtera, selamat pagi and a very good morning to all gathered here virtually. We are here for Cosmos of Curiosity Series 33. Thank you to Faculty of Intelligent Computing Unit, the respective officers, and broadcast control team here led by Mr. Shah for hosting this event. I'm Rabah, your moderator for this Cosmos of Curiosity Series 33 this morning. Before we begin, as is normal, I would like to introduce our esteemed speaker for this Series 33, Professor George McDonald Church, who will synthesize our understanding to grow just about anything. So let's welcome Professor George Church. His biography. George leads the synthetic biology at Wyss Institute of Biologically Inspired Engineering at Harvard University, where he oversees directed evolution of molecules, polymers, and whole genomes to create new tools with applications in regenerative medicine and bioproduction of chemicals. Among his recent work at the Wyss is development of a technology for synthesized faster, more accurate, and less costly than current methods. George is widely recognized for his innovative contributions to genomic science and his many pioneering contributions to chemistry and biomedicine. In 1984, he developed the first direct genomic sequencing method, which resulted in the first genome sequence, the human pathogen H. pylori. He helped initiate the Human Genome Project in 1984 and the Personal Genome Project in 2005. George invented the broadly applied concepts of molecular multiplexing and tags, homologous recombination methods, and array DNA synthesizers. His many innovations have been the basis for a number of companies, including Editas, which does gene therapy, Gen9bio, synthetic DNA, and Veritas Genetics for full human genome sequencing. George is professor of genetics at Harvard Medical School and professor of health. So George is professor of genetics at Harvard Medical School and professor of health sciences and technology at Harvard and the Massachusetts Institute of Technology at MIT. He's director of the U.S. Department of Energy Technology Center and director of National Institutes of Health Center of Excellence in Genomic Science. He has received numerous awards, including the 2011 Bauer Award and Prize for Achievement in Science from the Franklin Institute, and elected to the National Academy of Sciences and Engineering. Please hold your reins for the questions until after Prof. George has finished his talk. In the meantime, we cannot wait till the end. Please type your queries into the Zoom chat link. Thank you, and without further ado, a very good evening to George, and the platform is yours.
Thank you very much. It's great to be here. I will start sharing in just a moment. Everybody see that? Okay, so
okay, that's nice.
Okay, so
yes.
The topic is how to grow almost anything, which is actually the title of my course that I've been teaching for about a decade at Harvard, at MIT, and around the world. More about that later. But really, the topic is curiosity, and moreover, the research that we've done to try to satisfy our insatiable curiosity. I'll have some thank yous. Here's the thank yous for institutions that have helped us bring some of these things about. Maybe you've seen the abstract, but you'll get the idea in a moment. So we think of STEM or STEAM as science, technology, engineering, art, and math. I was fascinated by all of these and their intersections from an early age, and I've found it hard to drop any of them, including physics, chemistry, biology, behavior, ethics, and safety. And so I find ways that they can be combined in meaningful ways. And I'll keep referring in the talk to reading and writing, which is basically analysis and synthesis, because whenever you can do one, you can probably do the other, and it greatly enriches things. So I'm going to go through this narrative in a sort of mostly chronological order. The first three sections will just cover my high school education, my college, and my PhD very quickly, just to give you a feeling for where I came from, where the curiosity came from, and how one can maintain that multidisciplinary if there's a value to it. So this is starting around age 12. I was fascinated by orchids and Venus flytraps, and most significantly Luther Burbank, who did experiments on grafting hundreds of different kinds of fruits and cherries, for example. And I set out to do experiments on plant hormones that would cause extra growth. And so you can see the control on the left and the internodal distance, the growth. Clearly there was a significant effect of this gibberellic acid on the pea plants. This is same pea plants. And then I also did during that time in high school. These were independent studies, by the way. These were not courses. So I also synthesized organic compound, organic chemistry, cyclohexanone from cyclohexol, and then I did analysis using an infrared spectroscopy, which my professor had not done before. And then in math, I also did independent study during high school. In this case, Craig Bedford is the professor who let me out of the math class to work on symbolic calculus, differentiation, and linear algebra. And I used a teletype very similar to the one here that Bill Gates and Paul Allen were using. I've since then had many interactions with Bill Gates and Paul Allen. And we also used the kind of programs I wrote back then. We used that for metabolic optimization. And here's some of the code for the analytic differentiation. I didn't realize it at the time, but somebody at MIT had already done a thesis on this topic, but still it was interesting to do. So that's high school. Then college, I spent a huge fraction of my brief college career. It was only 2 years. I spent it in the lab, crystallography, X-ray diffraction of transfer RNA. And then so I worked with some pioneers in early computing, like the UNIVAC-1, which was used for the first time to predict presidential elections. And then Fred Brooks was the person who had the IBM 360. You've probably never heard of this, but this was the supercomputer of that era. And so he had two of them for his own department. He was chairman of computer science. And here's some of the math that I derived for a program that was in use for 3 decades called Corals. It did refinement of crystallographic coordinates and structures. And Fred Brooks wrote this book about the mythical man month, which has taken on greater significance. So this is what I looked like in 1973. I couldn't grow a beard quite yet. I worked with Sung-Ho Kim and Joel Sussman very closely. Joel was a postdoc at the time. Sung-Ho was assistant professor. And we just got together a few weeks ago for Sung-Ho Kim's 90th celebration, which was mostly a symposium on artificial intelligence and its relevance to proteins. And so I worked on transfer RNA during that time and learned a lot that I still use. We turned out that we were working with ribozymes. There was this pseudouridine that later turned out to be important for messenger RNA vaccines, and deaminase that turned out to be useful for CRISPR gene editors, and learned a lot about the genetic code, which have now made it into clinical applications. Okay, so that I learned a lot about tRNA in college, and then went on to continue to work on RNA for and the DNA encoded RNA in my PhD, and develop technologies for sequencing. So just as one slide on my PhD, I worked with Wally Gilbert was my professor for all of this, and we developed genomic sequencing method together in 1984. And then we applied it to immunoglobulin enhancers in 1985 with Anna Frusi, who was a graduate student at the time, and Susuna Tamagawa. And we also, Bernard Dijon was doing a long sabbatical in our lab, and he represented Piotr and Slonimsky, and we worked on things like guide RNAs and homing into nucleases. And really, the title of my thesis was Functions of Introns, because introns had just been discovered at the beginning of my thesis, and most people thought they were junk DNA, and my thesis showed 2 examples where it was not junk DNA. This is a rhinoceros sculpture that's in the front of the Harvard Biolabs. It shows a long history from 1962. Here's Wally Gilbert and Jim Watson, who ran our lab together for a while. And then this is iGEM. It's a international genetically engineered machines competition that's been going on since about 2004, and it involves 300 institutions worldwide. So also from Jim and Wally, of course, were pioneers in understanding the ribosome and messenger RNA and just very basic things that later led to the genetic code. And so I've been basically still working on transfer RNA for over 5 decades, changed from crystallography to the kind of molecular biology that Jim and Wally did. And one of the things we did is we decided we would try to recode. So there's 64 triplet codons, and we thought we would eliminate one by using the redundancy of the genetic code and move it into, let's say, one of the 3 stop codons, turn it into 2 stop codons. So the cell doesn't need the 3rd one, and then you can achieve 3 things. One is biocontainment. Another one is use of non-standard amino acids, and you can in fact use the non-standard amino acids to do the biocontainment. And I think the one that attracted me the most as we were planning this was virus resistance. We thought that in theory, if you changed the genetic code enough, you could get resistance to all viruses that either they're already in the lab or out in nature. And that seems to be the case. And in the process, we formed 2 companies that were interested in this altered genetic code or non-standard amino acids. And a series of papers, mostly in Science and Nature, thanks to these students and postdocs. In particular, I want to just show one slide on how we got to virus resistance eventually. It wasn't just changing one codon. We had to change 2 to get virus resistance and 1 more to get biocontainment. So this had all 3. It had non-standard amino acids, it had biocontainment, and virus resistance. And this was based on Syn61 delta 3, which came from Jason Chen via Adgene. And what we did was we did a simple swap of 2 codons out of 64. We swapped these 2 codons, which normally code for serine. There are 6 serine codons, and so we took 2 of them and moved them into the other 4. And so then those 2 weren't needed anymore, so we could knock out the serine tRNA that normally would be lethal. It wasn't lethal because we had recoded the whole genome, 18,214 codons by synthesis of the genome. And then we put in a leucine tRNA from a phage, which was very strong transfer RNA and overwhelmed any tRNA that the phages might bring with them. And to some people's surprise, a lot of phages do bring transfer RNAs, sometimes even all of the tRNAs with them that they need. But anyway, we found resistance to all the laboratory strains and mixtures of fresh environmental samples. So we think it's resistant to all viruses. You can see on the far right-hand side of this graph is undetectable titer after putting in, 10 million plaque-forming units per milliliter. This is thanks to Akos, Savannah, and Reagan, among others, that were co-authors on this paper. Okay, so then straight out of my PhD, I developed the so-called genomic sequencing method, And that was the first method where you could sequence DNA without amplification, without polymerase, without PCR, without cloning in bacteria. And then that led to genome projects. First the Human Genome Project, and then a variety of non-human genomes and personal genomes. And I formed the Personal Genome Project. And what we did was we changed the paradigm from gel electrophoresis to sequences of probing, imaging and then probing again and imaging again. And that led to genomic sequencing in 1984, and it led to fluorescent next-gen sequencing method in 2005, where we sequenced a bacterial genome to prove this. And this little image here is of a single cell where the same methodology for next-gen sequencing can be used on where every dot there is a single RNA molecule. And in situ, you can read out ACGs and Ts in 4 colors, and then the computer can recite the string of those so you can identify which RNAs are in which part of the cell or in which part of the organism. And that we made an instrument for next-gen sequencing that was just used by 3 of the top companies to get started with next-gen sequencing, selling instruments, and then we redid it for ReadCore to make a spatial 3D high-resolution imaging sequence in situ, and that can be used for DNA, RNA, or protein. I'll show you some examples as we go along. And we've also worked with Brooker Vutara, mainly in collaboration with Ting Wu's lab. So the 10x Genomics further developed our 3D spatial omics. Now the technology has been a very exciting ride. We've followed a kind of a Moore's law curve here in the dotted line. That's about how fast computing was going in terms of cost and other factors like that. And sequencing was going on a similar track. And early on, and when we first noticed it was on this exponential, we extrapolated, and it looked like we could get to a $3 billion haploid human genome. Hopefully, we hoped we could get it in a decade or 15 years. And we thought we could bring it down to an affordable, not $3 billion, but more like $300 in about 6 decades, 60 years. So that was a long time, but it was exponential, and it seemed about as good as you could do. Turned out it wasn't 6 decades; it was only 9 years. So it changed slope. So these Moore's law is not a law; it's just an observation that that happened to apply to DNA sequencing and synthesis, sequencing in red, synthesis in blue. And the synthesis is kind of hard to exactly there's a lot of different ways you can synthesize that uses combinatorics, uses natural processes. And so but you can get up into the order of 10 to the 17th or more base pairs now. And the price of these have dropped about 20 million fold in those 9 or 10 years, and we now have nearly perfect DNA sequence. We have an error rate less than 10 to the minus 10 for one genome, and that one genome happens to be from the personalgenomes.org. This was done by the Telomere to Telomere Consortium. Okay, so we'll pause for a second and talk about curiosity, and also our need to not just do basic science and curiosity, but at least have part of our mind thinking about what society needs, what applications you can get out of the technology while you're satisfying your curiosity. You can really, now the technology has gotten to the point where you really can do both. And so this involves a kind of ethics, which is you want to have technologies equitably distributed. For example, smallpox. The entire world is now safe from smallpox because it's extinct. And it was pretty safe when there was, first vaccine, but now everybody can get it. You don't need to update each year a new vaccine or a new drug. It's extinct. That's an example, but we're looking for others. And so there's some of the societal needs are, cardiovascular disease and diabetes and age in the wealthier nations. Poorer nations might have those, as well as TB, HIV, malaria, and nutrition. And definitely both have severe genetic inherited mutations. It ranges from about 3 to 6% of the population, depending on the level of consanguineous marriages. And there are other reproductive issues, psychiatric issues. And these reproductive, we can have too much population growth. It was alarming back in the 1970s, 80s. And now we have the opposite alarm going on, where we have if you look at these demographics on the far right, you have kind of an explosion of elderly people and an implosion of the number of young people to help take care of them and help the economy. So, but there's a whole variety of reproductive reasons for that. And then we have existential risks that we prefer not to talk about, but they're, asteroids, supervolcanoes, and so forth that could really cause major disruption of supply chains and chaos. And part of these things that we think about from time to time are we're encouraged. My wife and I, Ting Wu, started pged.org, which is all about the dialogue with the public, and Future of Life Institute was started. We joined that. So now stem cells, embryoids, and organoids has started my postdoctoral work with Gail Martin at University of California, San Francisco. And here, she was really one of the first people to establish embryonic stem cells and get them to grow. And that led to her professor getting the Nobel Prize for that sort of work, especially when applied to homologous recombination by Capecchi and Smithies. Anyway, I did a short postdoc with her. I learned a lot about development and got very interested in the cis and trans regulatory codes and the developmental codes that help us go from egg to adult to aging and death. And so here's some examples of stem cells, similar to the ones I worked with as a postdoc, but now fast forward to when I'm a professor, and these forming the same stem cells forming endothelia or excitatory neurons, just differing by one gene that we've introduced. Volker Buskampf and colleagues published the first of many papers in our lab on this in vitro epigenetic reprogramming from old cells to stem cells, and then from stem cells to neurons and many other cells. Now we use these neurons and neuronal progenitors and later brain organoids to as models for diseases, so we can test new drugs or test genetic effects. Here's one we were testing the genetic effects of Alzheimer's. So this is late onset Alzheimer's, and we found that you might imagine that to make a model of late onset Alzheimer's, you have to go for many years for it to develop as it does in nature. But we found that there were early markers that we could find that where the cases and the controls, the people with Alzheimer's and age-matched controls, had nearly identical transcriptomes, RNA, and these are hierarchical clustering of the RNAs. But you can see very clearly the difference in cases and control. It's just night and day. You don't even need a computer at this point once the computer has plotted them. You can see red and blue and red. And so this was highly predictive just from the behavior of the stem cells in terms of their transcription. And we could simulate part of this with as little as one genetic difference between ApoE4 and ApoE3. We did a similar set of studies on Alzheimer's disease I just showed you, Asperger's, autism spectrum disorder and bipolar disorder. And these in vitro differentiations and organoids have been very valuable for the kind of genetics I showed in the previous slide and for testing new drugs. And in fact, we have now a new drug applied to bipolar disorder that is based on previously approved, FDA-approved drugs in the forskolin family. So that was very valuable to have that. And you can see these beautifully engineered cerebral organoids. And this is Elaine Lim, who was the first author on paper from our lab on this. I'll take a brief side note on molecular electronics because it has something to do with sequencing and with neuronal systems. Here, one of we did two, I'll describe two molecular electronics. One was nanopore sequencing. I came up with this idea because I happen to know a little bit about single molecule neurobiology, which was published by Nair and Sackman. And I also knew about Coulter counters, which is a way you can measure individual particles of any sort going through a small channel. And the similar principles is that the conductance through a small channel could detect single particles or single molecules as ions would go through. And so this was at the beginning of the Genome Project, and it seemed like it was going to be a very expensive project, and I wanted to get something that was cheap and maybe had longer reads than anything that we were considering at the time. So we attached a protein motor, like a polymerase, to the DNA, and we could either have DNA going through the pore or being modified as it went past the pore. And so the through the pore ended up being Oxford Nanopore, and the past the pore was Genia, now subdivision of Roche. And so both of those are now commercially available. But it started in this 1987 realm with Department of Energy and this patent with Dan Branton and coworkers. So the second example of molecular transistors is where we literally have a transistor bridged by a long polymer like an alpha helix or a DNA double helix. So you have like any transistor, you have a source of electrons, you have a drain for those electrons, and you have a gate with a certain capacitance modulating the flow, the current going through the transistor. And we could make that gate very sensitive to single molecules, single DNA or protein molecules, or even small molecules like triphosphates. And so you could read off these spikes represent individual nucleotide triphosphates interacting with the polymerase. This was published in PNAS. So those are two examples of molecular electronics. For the nanopores, the Oxford nanopores here are on the ISS with astronaut Kate Rubins, who I talked to her while she was in space, and we've had many interactions since then. But she had four of these, really tiny. You get a feeling for how small these nanopore sequencing devices can be. She had four of them on the International Space Station. And the molecular electronics was partly inspired by neurobiology, and now we're trying to apply it to neurobiology. And we wrote a paper in 2012 about functional connectomics, connections of synapses of neurons in brains and outside of brains. And we proposed this to a number of granting agencies in the federal government, and Barack Obama's administration liked it enough that he initiated, and his team gave it the acronym BRAIN, which I particularly liked because the last two letters are innovative neurotechnologies. And I thought that was something we didn't really emphasize enough in the Genome Project, and here was a new project where we could really focus on the technologies up front. And then that funded, among other things, the IARPA Microns project, which was a $100 million project that Jacob Vogelstein and David Markowitz ran, and we were a grantee among it. And there were three groups that were grantees. two of them were doing electron microscopy, and ours was developing this fluorescent method, which is now quite common, and in fact is now represented at Eon Systems and E11 Bio. I'll give you an example of this. Andrew Payne and others worked on E11 Bio, and Michael Andreg on this. Part of this is helped by the super resolution sequence identification with multiple fluorophores, or expansion microscopy, which we worked on together with Ed Boyden in this paper, where we were doing nanoscale imaging of RNA using expansion microscopy. And then this is an example of a connectome of axons, dendrites, and cell bodies, neurons, and non-neuronal cells by fluorescence, where Eon Systems and E11 Bio are doing this. And so far it's been the largest connectome that I know of is in flies, but Eon and E11 are making good progress on getting the mouse connection for the whole brain done, and then they'll move on to human. And this is just an example where you can get information not just about connectivity, but functionality of the synapses, whether they're excitatory or inhibitory, which direction it's going, presynaptic to postsynaptic. And you can do that by imaging with antibodies or nanobodies, pieces of antibodies, mini binders that are labeled with DNA. And the labeling of DNA allows you to get multiplexing, and it allows you to get super resolution on the order of 20 nanometer resolution, which is at least 10 times better than you can get with conventional microscopy. Which then led us to protein design, somewhere along this path, where we used AI and a combination of machine learning form of AI, large language models and such, with multiplex libraries. So the machine language and the multiplex libraries were both powerful by themselves, but they were especially powerful together. And we started this back in 2014, where we were tackling a particularly difficult kind of protein. So some proteins are they're sometimes called rocks. They're kind of steady. They do one thing. They bind to one molecule. Very simple, highly soluble. But allosteric proteins have at least two conformations that differ. The binding at one site affects the binding at another site. And for example, the lac repressor family, which was one of the first DNA regulatory elements, it has a ligand that affects, a ligand related to lactose that causes DNA binding to change. And we changed that from being mostly responsive to this lactose-like ligand, IPTG, to having almost no recognition of IPTG and entirely focused on gintibiose or sucralose, which is a completely synthetic, non-natural artificial sweetener. So that was kind of before we were calling it AI, but it had a lot of the components, and in fact we tackled a project that was pretty hard to do even with AI. Fast forward a few years, 5 years, and we're doing in a big way, we're using AI to generate libraries of candidates. So we're not trying to get the AI to guess exactly the right capsid for delivering gene therapies, but to eliminate the ones where the changes are too neutral to have any effect, or too much breaking the function of the protein. In this case, we were doing the main gene therapy delivery vehicle, which is adeno-associated virus, or AAV, and we could get up to 99%. We could target a particular kind of neuron to 99%. We could get 280 times better brain targeting than anything previously, that we could avoid the liver, which is where a lot of gene therapies go, whether it's lipid nanoparticles or viruses. That's where they tend to go. And we could make libraries where you could change as many as 28 out of 28 amino acids. I'll show you that in just a second. And we did this by this loop of synthesis, selection in vivo, so we can inject 1 million different AI designs into a macaque and then find where they go. We can sample as many different tissues at whatever resolution we want, all at once from the same animal, and with 1 million viruses times dozens of different tissues and cell types. So this is thanks to Pierce and Eric, who then went on to form Dino Therapeutics and did these primate studies. And here's an example where with a naive model or a random model, we could hardly get any survivors. This is percent that are still functional viruses. We're trying to mutate a key surface domain of 28 amino acids on the surface of the delivery virus, and we could barely get 4 out of 28 changes and it would break. But here, with using logistic regression as one of our AI tools, we could get 27, 28 out of 28, 26 out of 28, very large libraries, which meant they were so different that they had a good chance of having a new function, a gain of function, which we could exploit to get different tissues. And Dino Therapeutics wasn't the only one that we launched at more or less the same time. Manifold Bio, that was doing a similar thing. Dino did it on capsids for delivery. Manifold did it for protein delivery. Nabla Bio was working instead of delivery, but on the actual therapeutic itself, focusing on G protein coupled receptors. Jura was doing AI, Helix Nano, and Lila.ai was aiming at all kinds of different science. These other ones were quite focused. And then we have a more general set of ethics and safety issues having to do with our improved DNA synthesis. So what I just described were ways that we would use these mega multiplex libraries, the MLML, to test, to find new functionality, new tissue preferences, new protein targeting, etc. And so, and when we came up with these new libraries in 2004, we felt it was important to accompany the paper that described the methodology with a critique of what the risks were and maybe some ways to mitigate those risks. And so, so this is the technology paper published in Nature, and this is the white paper that I wrote about how to avoid this by instead of just having voluntary consent but no surveillance. The idea was to get licensing and surveillance, and then that was resisted by the industry for about 5 years. But then they liked it enough eventually that it's near getting close to 100% adoption for any synthetic orders that are placed on the internet, as well as synthetic devices that sit on your desk bench. And then David Baker and I followed up on this 2 decades later, realizing that protein design was getting to the point where we had felt that the DNA was, and now it needed biosecurity, and it turned out it could piggyback on this synthetic biohazard proposal, and there's now an IGSC consortium and a secure DNA program development team that are helping us do this. So that's an example of what you can do proactively to anticipate problems that actually haven't materialized yet. And then, what we just talked about was this chip synthesis DNA that required something to go with it. There were issues for every one of these technology improvements we made. We would accompany it with an ethics and safety paper. Personal Genome Project was a reaction to concerns about privacy and sharing. You wanted both at the same time somehow. Mirror Life was an issue we raised in 2010 and just published a new paper about it recently. Human germline editing was a big thing at the beginning of CRISPR, and it was and it's already happened now. gene drives, this AI, ML I was just talking about, synthetic embryos, and other kinds of AI. I think particularly dangerous is general intelligence. And in fact, there's been a temporary ban. A lot of these temporary bans only last a couple of weeks. So de-extinction. This is a popular topic, even though for years people would keep asking us for updates on something, even though we had essentially no funding for it. Fortunately, we got funding in 2021, but it kind of started in 1990 with this book by Michael Crichton, who, by the way, was a student at Harvard Medical School. Dropped out to become an author, and he did very good work on screenplays, for example, for ER, emergency room TV series. Anyway, this Jurassic Park had two things that caught my eye. One was that it was a novel, and then it had, on page 103, it had DNA sequence. The first novel I saw that had DNA sequence, I wondered what it was. I knew it wasn't dino DNA, which was what it was supposed to be, because there wasn't a dino DNA at the time sequenced. And so we had very primitive tools for searching DNA sequences at the time, but I managed to figure out that it was not dino DNA. It was actually an E. coli plasmid that I had worked on in the 1970s as part of my PhD thesis. So that was kind of fun to see my old friend masquerading as dino DNA. And the other thing that caught my eye was this lysine contingency, which was the idea that if they escaped somehow, to skip the physical barriers, they would get caught by biocontainment, and in particular, they would run out of lysine. It turns out, however, that every living thing has lots of lysine in it, and so no matter what they ate, they would have probably satisfied their lysine issues. So I felt years later that the answer would be an amino acid like lysine that is not present in the biosphere. So the one we chose was BIPA, and I'll just show you that a little later on. The main idea, though, was de-extinction. Somehow you'd get a hold of the DNA and use a variety of tools to turn DNA in the computer, sequence it, put it in the computer, and then have the computer print it on new DNA, where you took this kind of degraded, mixed up, short read DNA and turn it into long read DNA, maybe with the help of a closely related species. Fortunately, mammoths and Asian elephants are extremely closely related. So anyway, print that into DNA, and then take the DNA and recombine it into cells, and then take the nuclei from those cells and use somatic cell nuclear transfer like you would like the sheep dolly and many other things since then. So anyway, and we were doing this, and we've now looked at many endangered and extinct species. It's mainly to get diversity back and to rewild ecosystems that are in trouble, at least with respect to our human sensibilities, what we think it should be, which isn't necessarily always right. But in any case, so in the case of Yellowstone, there's a nice case study that is somewhat debated, but anyway, it's that getting rid of the wolves was considered a good idea, and then later it was decided not such a good idea. When they were rewilded, then various herbivores upon which they preyed started staying away from the willows. Willows got to be full size. Beavers started harvesting the wood. Beavers made a rebound themselves, and they started making it a better place for fish and waterfowl and so forth. And so you can this was monitored as a function of years. And we wanted to make the elephants more like mammoths by studying the mammoth genome. We now have over 100 mammoth genomes that have been sequenced at fairly high quality. And so we have an idea of diversity of them, and then we know that they're very close related. Asian elephants are closer than they both are to the African elephant, which is a pretty quote. And the Asian elephants, these are not mammoths, these are Asian elephants that they like snow, and they'll make these giant snowballs, and they'll jump into frozen lakes and for a while. but they can't tolerate the minus 30, minus 40 degrees centigrade in the Arctic, while the mammoths could survive that for many months at a time. There's also a huge variation in tusk length. Here's some of the largest modern elephant tusk lengths in Africa. And then there is some like the ones up above, the Asian elephants, which have almost no tusks. Some of this is due to adaptation to poachers, to avoid poachers if you don't have tusks. There are animal models, like mouse models for tusks formation. Here's a mouse model for the woolly phenotype. Here's where it has relatively short, sparse hair. So these are examples of way we can debug traits in this short gestation period of 20 days for mice instead of 22 months for elephants. We also worked on EHV, more of that in just a second. So these are a point that here's a bison that I got to know while I was in Siberia last, and he was playing with a thin tree, but he didn't break that down. He was playing with it. While elephants, on the other hand, love knocking down gigantic trees as well as small ones, either to get the greenery at the top, the way giraffes might not, or they just like knocking down trees. And that's important in that to restore the Arctic. This is kind of a desert, partly because of the trees, to restore it to the highly productive grasslands, which sequesters carbon and allows for the cold air to reach. It's helpful to have the elephants, and then they would then be accompanied by a large number of herbivores that are faster gestation periods than elephants, and could maintain the grasslands once they're there. Colossal and our group have worked on EHV. The vaccine was developed in Paul Ling's lab in 22 and 24, both a subunit vaccine and a messenger RNA vaccine. And so this has been a killer, a big component to the endangered species, Asian elephants, and now was killing off maybe 20-25% of the newborns at the time of weaning, around 5 years old. So that's something that we want to do for elephants along the way. We have a variety of reproductive technologies that we're doing so that we can enhance the reproduction of endangered species. Here's an example of a pig uterus, which we're using as a test bed for some of the reproductive technology and new perfusion technologies. So here's how we moved into all these fields, kind of in the order that I've shown, immunology and virology, and applying those ideas to gene therapies and organ therapies. And a lot of these are now either approved or in clinical trials. And one of the things that worried me, I've already told you how I was obsessed with getting the price down, but for DNA and DNA reading and writing, and as a consequence, RNA and protein as well. But gene therapy looked like it was problematic. The price was not coming down by Moore's law or faster than Moore's law. It was going up. It's going up at about $1 million per dose per year. But then if you look at it a different way, so this is the cost as a function of time. And here's a plot of the cost as a function of market size. And you can see that for things that have very tiny markets, like most rare genetic diseases, the cost is in millions of dollars per dose. But if you get something that's very common, like the COVID-19 vaccines, whether they're viral vaccines or lipid nanoparticles, they have a single gene in each, and they can be in the $30 range per dose. So millions of dollars, $3 million to $30. And there's other things that bode, I think, give us hope that we're making progress on getting things out into clinical trials. I already mentioned the COVID messenger RNA, which is it's definitely formulated as a gene therapy, single gene, and its dose cost is not just because it was an emergency, not just because 6 billion people were using it and defraying the costs of the R&D, but it's also it could be delivered in fairly low doses compared to other gene therapies, intramuscular, and then amplified by the immune system. And so we're aiming for ways that we can reduce doses. Baby KJ was very encouraging. Here's a child that was born with a serious Mendelian disease. 7 months later, with FDA approval, the child was fixed. So that was a big decrease from the normal 10-year or more approval process of 7 months. And we're also, the FDA and society is getting used to the idea of polypharmacy. We have multiple drugs that either deal with multiple different ways that the disease can come about, or multiple ways that you can become resistant to the drugs. And polypharmacy is mainly cancer and infection, but I've argued for years that age-related diseases or aging itself has multiple causes and multiple tissues, and so it probably will involve complex polypharmacy. So here's an example showing how much we know. I mean, there's a lot we don't know about aging, but this is some of the pathways involved, and many of these have now been tested in therapeutic form, and a significant fraction of them have a survival curve that shows a shift to the right, which is indicative of a longevity effect. But we don't go to the FDA with longevity effects. We go with reversal of serious diseases. And one of the ways that de-aging can work is caloric restriction. Another way is when you transfuse young blood into old animals, it causes rejuvenation. And so if you can deliver to the liver produces most of the molecules, protein molecules in blood, or to the bone marrow, which delivers most of the cells. And so we went through, and we meaning Noah Davidson, as a postdoc in my lab, tested 48 different gene therapies in a very short period of time, very surprising to me, and then chose 6 of them, two sets of 3. One of the sets of 3 was this ATF, alpha-klotho, TGF-beta receptor, and FGF21. And the other one was OSK, down at the bottom here, which were Yamanaka transcription factors. So these were all secreted factors, and these were all nuclear factors, because you can see here it's the extracellular medias at the top, and then the bilayer, and the cytoplasm, and nucleus, mitochondria, and so forth. And so this is, and we've continued to explore almost everything on this page here. Here's an example where we did all combinations of these 3 out of the original 48 gene therapies. We decided to focus in on all the combinations of them here, color-coded, and we used a variety of tests like to accelerate the aging or make it more emphatic. We would give them a high-fat, so-called Western diet, ad lib, and then with the purple and red treatments, gene therapies, you could see that they would come down in weight fast and then plateau. And this has implications for a variety of diseases, including diabetes. Here's a direct test for insulin tolerance, which again shows those same winners improving in this insulin tolerance test. Or you can damage the heart or the kidney and see how quickly it grows back. And this is something that youthful animals can do, and we could get it to go with these gene therapies. Now here's a different set of 3. So this is mostly the ATNF, and these are the OS and K genes. And here it was administered at the point where half of the animals were already dying from old age, normal aging. That's this black control line. And then injecting this intravenously, these 3 genes as all in one viral capsid that kind of packed in 3 genes into one capsid. And you can see a very significant shift to the right in the blue line, where the treatment with the 3 genes relative to the control, which is this is an example of using a triple gene strategy. And here's not we now got 7 or 8 different age-related disease indications, so we're not just testing biomarkers, we can actually test this out on real diseases that the FDA can approve without having to convince them that aging itself is a disease. It's just take these age-related diseases. This is thanks to Noah Davidson at Rejuvenate Bio, where we now have these kinds of gene therapies going into clinical trials, both for veterinary use in pets and in human as well. Here's a different project focused not so much on gene therapy for aging initially, but for making new tissues epigenetically by introducing transcription factors like the OSK, but these are now, OSK takes you to a younger age, and these are moving it to older, more differentiated stages. And so, for example, here we're making oligodendrocyte precursor cells, which are capable of differentiating. They've already differentiated from pluripotent stem cells into oligodendrocytes. They can wrap around axons, such as you have accelerating neuronal messages in your white matter in your brain. And we showed that we could restore a lot of the functionality to animals that had an autoimmune demyelinating disease analogous to multiple sclerosis. We can restore that function. This is thanks to Alex and Peristu, who published this paper in Nature Biotech and then formed GC Therapeutics, where they now are applying it not just to these autoimmune diseases, but to age-related diseases as well. Now, one category of disease that not necessarily these ones that I've been talking about so far, but a wide variety of heritable diseases, very severe Mendelian diseases, very often caused by a single gene or a very simple genetic pair of genes. And this sort of disease can cause very early death and pain for the entire family. 3 to 6%, depending on the population. And so there are various ways we can address this. One is carrier screening premaritally, in vitro pre-implantation and in vitro fertilization, and then later stages. The advantage of IVF plus whole genome sequencing. So this is something that Noor Siddiqui, who is founder and CEO of Orchid Health, pioneered, which is really high quality genome sequencing. There's just a few cells at the 100-cell embryo stage in vitro. Pluck those cells, keep the embryos frozen, and then you can catch de novo mutations, which is nearly impossible to catch so far with carrier screening. Also with carrier screening, if you find out how you're dominant, that there's no real solution to that other than IVF or later gene therapy. And you can even reduce miscarriages. A lot of people that are in the IVF clinic have male or female or unknown causes for miscarriages or for infertility. And you can also reduce the number of cycles. So one of the major problems here is the pain and the cost and the inconvenience of these. The disadvantage is the cost, the pain, and the multiple cycles for each embryo. But if you can screen with whole genome sequencing, you can reduce the number of cycles, pick the best embryo, the one that's least likely to cause a miscarriage. Furthermore, we can reduce the pain additionally by and lower the cost by reducing the hormones, which produces more immature eggs, but then we can mature those eggs in vitro, so-called IVM for in vitro maturation using granulocyte-like cells, which we get from pluripotent stem cells. And so this has now become the first FDA-approved clearance for a therapy to enter phase 3 clinical trials, and it's helping women with in IVF clinics already. Dina Turner is the co-founder, and Christian Cram and Merrick Smila were instrumental in starting these two companies, Gamito and Ovell, to deal with this kind of problem. Now, Lu Han Yang was a graduate student and postdoc in my lab, and she helped establish CRISPR as a technology, and then we were invited by some of the pioneers in the field of xenotransplantation, transplantation from animals to humans. We were invited to apply CRISPR to the problem that they had once thought was as little as one gene, and then by the time we were done, we realized it was at least 69 genes, and so we published a series of papers and started this eGenesis, and it was really aimed initially at kidney and now liver and heart, in that there are hundreds of thousands of people on dialysis, but a tiny fraction of that, maybe 1/20th of that are getting transplants per year. And the dialysis is really not their, it's better than dying, but it's not their choice if there's any alternative like a kidney transplant, because they spend 3 days a week, 4 hours per day, and it still leaves them in a brain fog a lot of the time and limits travel and so forth. Anyway, so we did 69 edits. 3 of them were very severe sugar glycan incompatibilities in blood complement, clotting, innate immunity, inflammation, and so forth. So that was 3+7 changes. And then we knocked out all of the endogenous retroviruses, which the FDA was concerned, rightly, that they would, they were very aggressive retroviruses that would be secreted from essentially every organ of the pig. And since the recipient human was immune suppressed, as are all human-to-human transplant patients, this would be a possible recipe for zoonotic disease development. And so we tackled that first, and we found it was actually fairly easy to knock out all of the retroviruses, and they didn't return even though these engineered pig embryos were implanted into a mother that still had the retroviruses and they didn't reinfect. So the way we knocked out the retroviruses retained the envelope and protected them from reinfection. So here's one of the first surgeries where a pig kidney is going into a patient named Tim Andrews. He ended up being one of the longest stable xenotransplants, and he made it 270 days and then got a human kidney. And he was very happy both with the pig kidney and the bridging to a human kidney. And we want to do this not just because there's a deficit, an inadequate supply of human organs, but we'd like to make enhancements that really requires germline engineering, which we can't practically or ethically do on humans. And some of these enhancements we'd like to be able to store them in the cold, which is not really feasible now. We'd like to have them be DNA damage resistant for compatibility with chemotherapies, for example. We've already talked about immunity and pathogens like the retroviruses, but we'd like to make that more general so that they don't need immune suppressants. And cancer and senescence are things that have been shown we can improve these in animal models like mice. We'd like to transfer these to pigs and then use those pig organs in humans. And everything up here has been shown either naturally in animals or with synthetic biology in animals. So, for example, there's a Siberian salamander that can survive minus 55 degrees centigrade for a long time. And I just want to end there and open up the Q&A now. I want to say Thank you to my lab here. This is most of my lab. And then also thank you to the alumni from my lab who got together every now and then in local groups, or here's one big group of 200 of them that showed up. And the Personal Genome Project, which involves volunteers that are willing to share their medical records and their genome freely in a way that can be used commercially and can be used without worrying about de-identification. And I'd also like to thank the people who helped me teach this course at Harvard and MIT and now Wellesley, and 15 global labs and 1,500 students worldwide how to grow almost anything, which was kind of the theme today. So I will stop sharing and open it up for Q&A.
Thanks, George, for the enlightening look into the whole spectrum of genomes and DNA stuff. If there's anyone who'd like to ask questions, we are open for anyone in the chat in the Zoom. If you're ready to ask questions, you may ask George. Else, we'll start reading.
Yeah, Prof, I have one question for Josh. Yeah, you mentioned about the exponential graph of read and write DNA. I don't know whether I missed your information. Do you think that the trend can continue or approaching a fundamental limit?
Which trend is that?
In your graph.
Oh, the sequencing and synthesis graph?
Yeah, that the straight line, right? So whether this will be continue, right?
I think
approaching a fundamental limit.
we need to mute. Somebody needs to mute. Anyway, they may continue. It's very much like the Moore's law for electronics, is that we may change exactly what we mean by it, or there may be another revolution. I think there's easily another 5 orders of magnitude that we could bring down the cost of sequencing when it's applied specifically to things like multi-omics and in spatial omics and connectomics. For synthesis, I mean, there are demonstrations that we can do ridiculously large synthesis projects, but what we're not so good at is completing, a radical redesign of a genome, and that's so we're limited to bacteria so far, and that even bacteria take a decade to synthesize the genome. I think we're going to see a big jump in that in the next couple of years, where we can get from a bacterial genome in a decade to a mammalian genome in a few days. So keep your eye out for that. I mean, I don't know exactly when that is, but I think that's an example of how those curves are going to keep going. Maybe 5 orders of magnitude.
George, I have one. Why has cancer been so difficult to battle?
So cancer is a little bit like infectious diseases in that they're both evolutionary diseases, that they're not like cardiovascular, which typically is what it is. There actually is Darwinian processes involved in cancer. It's some of it has been is genetic, and there's things that you can do about that. A lot of them are environmental, and most of the real breakthroughs initially had to do with prevention. Eliminating carcinogens, smoking, UV, etc. It is a tough problem. I think it's tougher. Now, a lot of the cancers are age-related, not all of them, but a lot of them are age-related. So that might be that if you can solve aging, you will have to make a big road into cancer as well.
So the environmental catalysts are the difficult part to solve.
Right. Well, I mean, we are getting better at knowing what those are. We've got a whole new category of them, which is microplastics, which may or may not be related. But yeah, I think we're getting better at imagining what they are, detecting them, and then correlating them, and then eliminating them.
Okay, anybody else want to ask any other questions? If
There's
not,
some in the
we'll go to the
chat
chat.
that I can answer. There's
Yeah.
one about semiconductors and the metaphorically equivalent in biological systems, which have been described as like, repressor systems, and those are very slow. So the semiconductors can work on the order of gigahertz, while the biological ones on the order of hertz, or millihertz, kilohertz. What I was talking about by molecular transistors in the two examples I gave include molecular transistors that operate at the speed of light. So, for example, the second example I gave are in every way conventional transistors. It's just that the gate happens to be single molecules, and so those could operate at the speed of light, and they could be made compatible with biological systems. But it's very early days. That was one of the first examples of a large-scale integrated CMOS circuit, which had molecular gates. A good question. It is true that most talk about synthetic biology is talking about very slow circuits, but I gave another example, a fast example.
I think still have the question. I think Prabha is going to. He's in hospital right now. He have an appointment with the doctor, so I think I will take over from here. So
Right. Thanks, George.
yeah.
Hope to talk to you some other
Hope
time.
we get well. Yeah.
Yep. Thank you.
Yeah.
So I think they have a second question from Prabha, I think actually. It's about the large language model of AI processor used by someone to visually screen new stuff because you mentioned about the libraries of the genome and protein.
Yeah. Most of those libraries are fairly specific, but they can be reused. For example, our transcription factor library, we've used again and again for multiple different tissues, and now not just for tissue specificity, but for aging. So, it's some combination of libraries that are very specific and libraries that are more general.
I think the second one is about the semiconductor technology.
Yeah, so the second question about the semiconductors and transistors is that the ones that we deal with that are manufactured to very high precision with silicon that is, less than 1 part per billion contaminated with non-silicon, and they're incredibly precise in their positioning, 2 nanometer technology. While biology is very irregular, is the point. And the idea here is that biology has the ability to do error correction. We don't know what the upper limit of that error correction is, but it's on the order of 10 to the minus 10 for polymerase, for example, and polymerase plus error correction. It's built into the biological systems. But here, if we're talking about making molecular electronics that computes at the speed of light, then we are competing more directly with semiconductors. And this is just a challenge. It's like a newborn baby. You don't know what it's going to do. But one thing that biology can do that we can't do with large-scale integrated circuits is evolution. I mean, we can, in biology, you can make, instead of one circuit and test it heavily, we can make 1 trillion circuits and let them compete with one another and pick the one that works the best. So you can build into that fault tolerance, error correction, and so forth. I'm not making a bold prediction that it's going to be better than conventional electronics, but it's different enough that it's worth trying this fusion of evolution, AI design, and molecular scale transistors. But it's totally a great question. It's by no means guaranteed.
Yeah, the next one is from Chai Ming Yu. Things about AI and then the health of the people who live nearby the AI data center.
So it ends with, is there a way to ethically use AI for future research without affecting the global climate and resources? It's a terrific question, and it is one of the motivations for why we're looking for biological alternatives to GPUs and so forth in the previous questions, because biology is extraordinarily low energy consumption. We can get very close to the thermodynamic limit for irreversible computing, and there are some biological systems that do reversible computing, which are even more energy efficient, but they're not available now. I would say that a lot more of the energy consuming hardware and software is focused on general intelligence than it is on research. The research demands for it are relatively modest, but time will tell. I think it's an open question, but I think overall that what we get out of the artificial general intelligence is a little bit riskier and a lot more energy consumptive, and there will be innovation there, and there will be a lot, hopefully, a growing amount of economic analysis focused on the trade-offs.
Yeah, the next one is from, I think, question from Prabhu again. So how much of the human genome has been discovered and has been functionally categorized? And how much of the degenerative human disease could we currently treat utilizing gene therapy? Would the recovery be equivalent to the person going for surgery or more akin to taking vaccines?
So these are soft numbers that, there's a great deal missing interpretation of what the function is in almost every genome, not just the human genome. Even very well-studied organisms like E. coli, bacterium, there's 20-30% of the proteins don't have function. And even the ones where we have assigned a function, you might say, oh, this looks like an esterase, but what kind of esterase? And what does it do exactly? So, and is there a second function? Some proteins have more than one function. So that's an open question, but there's a lot that one can do with engineering without complete understanding. I will say boldly, for example, the development of the first smallpox vaccines in the 1700s was done without any knowledge at all of virology or infectious disease and immunology. They just didn't, they were invisible. The immune system and the viruses are invisible. Nevertheless, the protocol that was developed persisted for 3 centuries, and it's basically the one we use today. So I think engineering is the art of doing whatever you can with whatever you know at the time. Nevertheless, we have tools now that are helping us systematically determine functionality in a variety of genomes. And sometimes if you learn it in one genome, you can, it gives you a hint as to what to look for in homologs in other genomes.
Yeah. I think that the last one is about, whether knowledge and technology, we can rewind the T-Rex of many the mammals. I think the one that you have. Yeah.
So T-Rexes would be very difficult to do via DNA because we have not yet recovered any DNA from anything older than 2 million years. And the T-Rex is quite a bit older than that. But there's no proof that 2 million is the limit. In fact, for a while, people thought the limit was 8,000 years. and it just kept getting older and older until finally we found 2 million so far. But there could be some technology that could, for example, be able to sequence DNA in glassy minerals or something like that without extracting. The mammoth, though, we have hundreds of really high quality genome sequences, and so really what's lacking is the ability to synthesize those genomes and test them. Or if in addition to making kind of a perfect copy of a genome, you might want to change it in interesting ways, and you might even want to determine what's the minimum number of changes that you can make in order to recover certain traits that you like, avoiding traits you don't like, and so forth. For example, the trait of EEHV sensitivity you might want to get rid of, or you might want to get rid of, the tusks for certain applications. So, T-Rex and the mammoth are neither are done yet, but the mammoth is closer to being able to do a pure DNA. The last question is on mirror biology. This is something that has been described as a large threat by some of my colleagues. I did sign up to be author on a paper bringing this to people's attention, partly because I like to, I think that dealing with issues long before they become catastrophic or even of concern is good, because if you wait until the last minute, then you tend to make rush decisions, which are sometimes the cure is worse than the disease. The fix, the technological fix, is worse than the problem. I think we do need more study of mirror biology, both because it's very useful. A huge category of drugs are based on mirror biology, and antibiotics, surfactants, and so forth. But we need to see if these things affect. We might need to protect ourselves from mirror life on Earth that we haven't discovered yet, or from other planets, or from people that are wishing to do harm in a big way.
Josh, I have one question. Since I think your works bring together the genetics, engineering, computation, and chemistry, and physics. So do you think that the future of biology will require scientists to become more interdisciplinary, or we will eventually need a completely new discipline?
I think for a while, for many decades, it's been great rewards for people who could specialize and really, get the reward of studying a system better than anybody else. And I think that is still true, and that will be true, that there'll be greater and greater specialization required, both in the discovery, the engineering, and the medical practice. All of those will have to have components that are highly specialized. You also need to have generalists, and always have. For example, the general practitioner or personal physician has to know at least who to refer you to. And I think the same thing is true in technology development and in basic scientific research. But what's different is that now we really have a new set of tools that really enable you to practice multiple fields, as I've demonstrated in this talk. Some of that's due to the incredible help we have from AI. Some of it's due to just the conventional automation had such a big role, and also the ability to outsource some of your tasks to companies that will do it as work for hire. So there's a lot of ways that you can integrate multiple technologies in one person or in one small team. I think it helps to have, if you're going to do interdisciplinary work, it helps that your team not be entirely composed of highly specialized disciplinarians. Phase 3 and personalized cancer vaccine. I think a lot of us knew that something like that was coming. But it really is a very exciting moment. I think I think I share the excitement that many people outside of Moderna share. It's following. It's a kind of a pattern that some of the companies I work with are following, which is the N of one. Is that you could have a quicker approval process. The whole point of the large FDA-approved clinical trials is when you're going to be applying one strategy to a large population, and you want to see using statistics whether it's going to actually hurt or help that population on average. But if it's an N of one, there's no way you can get a large population. And so, ironically, the approval process could be much faster and cheaper. And with cancer, we've been talking about this for quite a while, is the possibility of getting neoantigens, antigens that are specific to that person, just because many cancers are highly mutagenic to the whole genome, not just the parts that are highly selectively advantageous to the cancer. And so those produce new antigens, and those can be harnessed for making vaccines. So I think it's a very encouraging for anybody that's developing any kind of messenger RNA drug, and for anybody that's developing any kind of N of one personalized cancer vaccines, whether or not it uses messenger RNA. So it's a boost to both those fields in the intersection. Very exciting.
Yeah. I think the last one, Josh, do you have any advice for the young scientists, researchers who are very curious about, about the new kind of research? Do you have any advice for them?
Yeah, so I mean, this is a frequently asked question. If I'm hearing it right, the advice for especially young people, but people of all ages that want to change fields or add a new field, I think it's a really great time to be a scientist. It's a pretty good time to change fields. We've got AI. You might evaluate whether AI is your friend or your competitor in your particular field. Is anybody's guess for some of these things, but I think it will be very helpful for people in some fields, and for others it will replace them swiftly. And it's just like a lot of other technologies in the past. A lot of simple automation that didn't involve what we call artificial intelligence has led to people seeking new jobs. So, anyway, it's a good time. Technologies are constantly improving so much that it becomes a level playing field where an outsider, someone coming in, can be competitive with the insiders, because there's a new technology that really nobody knows other than the lab that developed them, and even the lab that developed them doesn't necessarily know them. And some of these technologies, are relatively easy to pick up, especially with the help of AI to help you pick it up.
Yeah. Okay. So I think we have end of the time. It's about 10, almost 10:30 in your the night in your place, Texas.
Right.
So thanks again, Josh, for the very wonderful sharing session. I think this is probably new for Malaysia to see the huge collaboration about this kind of research, and I hope they will be impactful for everyone who attend this session. So for the end of this session, hopefully all the participants can you switch on your camera? So we want to take photo with Josh. Yeah, cameras. Shah, are you ready to?
Okay, boss. Open camera, please.
Yeah, is it okay, Shah?
Okay, kejap.
Okay, so we have the link of the attendance. So okay, so thanks, Josh. Thanks again for the
Thank you all.
yeah.
I
Yeah.
wish you great success
Yeah.
in whatever direction you take, and biotech is a very good direction. So
Yeah. I've planned for you for the next year with Jennifer, hopefully, with the
yeah,
special series dialogue.
okay.
Yeah.
Take care, everybody. Good
Yeah.
luck.
Thanks, Josh. Bye. Bye.
Bye.